Hi, everybody. Can you all hear me?
Yes.
Thank you. This is our next to last lecture. We're going to finish talking about cancer. And then with time remaining, we're going to delve a little bit into systems biology, which is different from what we've been talking about most of the semester. So we're going to finish up cancer. And like I said, then we're going to talk briefly about systems biology, which I think is a very timely topic, actually. We talked about gatekeepers, proteins that ensure that the cell cycle does not progress if there is something wrong. One of the most important ones of the gatekeepers is a protein referred to as P53. It monitors the cell for DNA damage. So sunburn, P53 is going to activate another protein, P21, which is an inhibitor of cyclin CDKs. It's going to prevent progression through the cell cycle. In the absence of this functional cyclin A, CDK2, you don't get phosphorylation of retinoblastoma. The complex, retinoblastoma, stays complexed with the transcription factor E2F, so it prevents progression through the cell cycle. E2F is not released. It does not go on to activate transcription. So the enzymes for DNA synthesis do not get produced. And I can never make this little box big enough. The cell cycle is halted prior to movement into S phase, the synthesis phase of DNA replication. So one of the gatekeepers, P53 is very important. In roughly 50% of all cancers, P53 has been mutated so that it is no longer functional, can't assume its gatekeeping role. So cells that are damaged, DNA is damaged, can continue through the cell cycle. And if you recall from one of our previous lectures, lost my train of thought. Cancer, one of the characteristics of cancer cells is that they are pretty much immune to mutagenesis, to X-rays and things like that. It's because the vast majority of them, P53 is no longer functional, and so cells can progress even though their DNA is damaged. Very important protein. It's one of the tumor suppressor genes. As long as you got one good copy, you're okay. But if you lose both of them, you're in trouble. Loss of checkpoints, however, is not sufficient to propel the cell through the cell cycle. You need other events to occur to initiate the cascade. Typically, protein kinases are the enzyme that initiates the cascade of events. You need external and or internal and or external signals to start the cascade leading to cell cycle progression. We've been talking much of the semester about programmed cell death, otherwise known as apoptosis. Cells not needed in development, such as the epidermal cells between our fingers, toes, they commit programmed cell death, apoptosis, or damaged cells, which is not programmed in the sense it's not part of the normal developmental process, but it's part of the surveillance mechanism that our body employs. One of the first things that happens is that the DNA, the nuclear DNA, gets fragmented, organelle structure becomes disrupted. Cells lose their shape and become somewhat spherical. And most importantly, cells break up into apoptotic bodies, which get consumed by scavenger cells. This is very critical. Apoptosis is an organized way of eliminating cells that are no longer needed or could cause damage. This is an illustration of apoptosis. You can see the cell with the various different organelles is intact. It starts to undergo apoptosis. You see condensation. The DNA is starting to be fragmented. You get fragmentation of the entire cell and forming these apoptotic bodies. And the most important thing to remember about these apoptotic bodies is that all the cellular contents are surrounded by cell membrane. This is not simply a release of cellular content into the environment, but it's a controlled process whereby everything inside the cell is in these apoptotic bodies with a membrane, bound by a membrane. The initiation of apoptosis involves cysteine-containing aspartate-specific proteases, otherwise known as caspases, for obvious reasons. It's much shorter. The caspases are a family of proteases that cleave the target proteins, which in turn initiates the cascade, resulting in degradation of DNA. The caspases exist as a zymogen, which are an inactive protein needing cleavage in order to activate. So cells, the vast majority of our cells, have these zymogens present all the time. They're simply awaiting the events that cause cleavage to activate the protease and initiate the cascade that leads to apoptosis. The triggers can be intrinsic triggers, reduction of growth factors, P53 sends up signals, cell damage, things like that. It can also be extrinsic triggers, Fas ligand, tumor necrosis factor, various other things can also initiate the process of apoptosis. There are various insults to the cell that can result in leakage of the mitochondrial contents, which is a very toxic event. Cytochrome C, which is involved in respiration, binds to a protein, APAF. APAF stands for apoptotic protease activating factor. This complex of APAF and cytochrome C complex binds to the initiator caspases, and it initiates the cascade. You can inhibit apoptosis in part by preventing mitochondria from bursting. And frequently you'll hear somebody seeking publicity talks about they've come up with a cure for cancer. And many times it involves initiating disruption of the mitochondria. The problem is we know how to kill cancer cells. The key is killing the cancer cells without killing the patient. There's literally probably millions of ways to kill cancer cells. But the problem is it also kills normal cells as well. And that's the challenge with treating cancer. Yep, you can also bind up APF so that it cannot bind with the cytochrome and initiate the process. So a couple of ways of inhibiting apoptosis. This is a YouTube video that I think helps to clarify what's going on in apoptosis. And again, I'm not going to show it, although I may at the end of the lecture today show one on related to systems biology that I think is very informative and very useful. Possibly one of the best YouTube videos I've ever seen. Apoptosis is not the only way that cells die. The other option is necrosis, and necrosis is by far the more damaging event. Again, it's cell death due to damage, but it's a form of damage that does not result in the initiation of apoptosis. Holes in the cellular membrane end up causing leakage of the contents of the cell to the surrounding environment. Lysozymes get emitted, leaked into the extracellular matrix, which is what holds organs together. Causes inflammation of surrounding cells. Apoptosis, as I said, does not cause any leakage. The apoptotic bodies are surrounded by a membrane, so they stay intact until they get consumed by other cells. So there's no inflammation. Yeah, membrane remains intact in apoptosis. Here's a slide showing the difference between the two. Necrosis on the left. Cells are swelling up, taking in water, causes the cells to rupture, and now they're dumping all the cellular content, including all the digestive enzymes and everything, to the surrounding cells, which causes inflammation. In contrast, on the right, we have apoptosis. Cell also starts to change. In this case, they're actually expelling water, and results in blooding, if you will, or budding off of cellular components, but they're all contained within a membrane. So there's no leakage of cellular content into the surrounding cells, no inflammation is caused, and eventually they get consumed by phagocytosis. Much better mechanism for eliminating cells that you need to get rid of. Talk briefly about signaling. Ligands, which are the proteins that bind to cell surface receptors. They come in actually 3 different groups, if you will. There are the juxtacrine ligands, which are membrane bound. They are not diffusible, they never leave the membrane, so you have to have cell-to-cell contact in order for juxtacrines to initiate a signal cascade. You have paracrines, which diffuse locally, or about a 20 cell diameter. They will bind to receptors on other cells. And then you have endocrines, or hormones, that are produced in, say, the thyroid and migrate throughout the body. So 3 different levels. Juxtacrine requires cell-to-cell contact. Paracrines diffuse over a fairly short distance, diameter of roughly 20 cells. And then endocrines, which can diffuse throughout the body. All are ligands that interact with a receptor on the surface of membranes and initiate a cascade of events inside the cell receiving the signal. The binding of ligand to receptor initiates signal transduction cascade. You're getting information from outside the cell to inside the cell, and the cell is responding appropriately. Can be used to regulate cell cycle, to induce or inhibit apoptosis, can also result in cell migration, changes in cell shape, can initiate transcription of genes. A whole variety of different things can be initiated through this kind of signaling. You can have positive signaling, mitogens, which are usually paracrines, or growth factors, which are diffusible over greater distances. Many of the mitogens activate tyrosine kinases, Again, we see kinases over and over and over again when we're talking about developmental events. These mitogens activating the tyrosine kinases result in expression of cyclin D proteins. We've talked about how they play a critical role in progression through the cell cycle. Some examples: epidermal growth factor, EGF, epidermal growth factor receptor. The EGF is the ligand, EGFR is the receptor. These names are historical remnants. These factors, virtually all of these factors, don't just act in the tissue that they're named after. It was where they were first discovered. Epidermal growth factor acts in a lot of different tissues, not just the epidermis, but that's where it was first discovered. Sort of a historical legacy, if you will. You can also have inhibitory signaling. TGF beta is a ligand that binds to its receptor, activates receptors serine threonine kinase. Cascade blocks phosphorylation and inactivation of the retinoblastoma protein. You may recall that to inactivate retinoblastoma, can no longer bind to the transcription factor E2F, requires phosphorylation of the retinoblastoma protein. Upon phosphorylation, E2F is released, migrates to the nucleus, initiates transcription, leading to the S phase of the cell cycle. So some of these inhibitory signals actually prevent phosphorylation of retinoblastoma, so the cell doesn't progress through the cell cycle. E2F remains inactive. No progression. No progression of the cell cycle. Positive apoptosis signaling. Critical quantities of immune cells, for example, become too great. FAS, the ligand, binds to the FAS receptor. In adjacent cells, the cells to be destroyed. FAS receptors come together, much like we talked about with HIV, recruiting other receptors to form a complex. In this case, it's a trimer. It indirectly activates APAF, the factor involved in initiation of apoptosis, which activates the initiator caspases, and the cascade begins for the cell to commit apoptosis. Programmed, controlled cell suicide. As I've told you several times, the default state of the vast majority of cells is to be primed or prepared to enter into apoptosis. They have the caspases present in the form of a zymogen. They're present all the time, and they're awaiting signals to cleave and become an active protease. So cells are primed to commit apoptosis. Because when a cell goes bad, you want to get rid of it as quickly as you can and ensure it doesn't progress through the cell cycle, producing more defective cells. In order not to initiate apoptosis, most cells at least require an external signal to remain alive, so-called survival factors. Some of them are mitogens. Others are not, but many of them are mitogens. Mitogens initiate mitosis or allow for mitosis. Oops. Okay. You already seen about my dog, our son's dog from China, the rescue dog Peter, and how he's a temperature sensitive mutant. So this is going to end our discussion of cancer. So now we're going to move to something we really haven't talked very much about, and that's systems biology. thus far, what we've talked about is largely referred to as reductional biology. Just like Gregor Mendel, he was successful coming up with his observations and insights and hypotheses by reducing genetics to as simple as possible a system. He looked at one pair of genes at a time initially, ultimately went to mating with several different pairs, but initially he made it as simple as possible. And the vast majority of what we've been talking about, and the vast majority of genetics and our understanding of developmental biology, all came from reducing things to as simple a process as possible. Typically, one mutation in a critical gene at a time. You will accumulate mutations in other genes in the pathway, but it's still focused on one gene at a time. Reductional science, reducing it to the simplest system possible. Systems biology, on the other hand, looks at integration of information across the systems, and it's much, much more demanding than reductional biology. Vaccination, this is where I said this is a fairly timely lecture. Folk wisdom in Europe, milkmaids never got smallpox, or if they did, it was extremely mild. What they did get was cowpox. Cowpox is a very related disease. It causes boils on the hands of typically females that were milking cows that were infected. And so they got the disease by milking the cows. And it resulted in a very relatively mild disease, easily recovered from. Smallpox, on the other hand, very deadly. Several monarchs in Europe died of smallpox. A physician in what is now the UK, Edward Jenner, suspected that there was a link, that somehow being exposed to cowpox resulted in immunity to a serious infection by smallpox. So he selected a young boy, a volunteer, which I use that word sarcastically, with increasing concentrations of cowpox pus. So he started off with a relatively small concentration and increased the concentration over time. Then, at some point, he felt probably the child should have immunity to smallpox, so he injected him with pus from smallpox. The child did get sick, but he rapidly recovered from the infection. So it did confer resistance to infection by smallpox. It was the first vaccination. Here you can see the willing volunteer doing his best to escape, with his family surrounding him as this physician is injecting him with cowpox. These kind of experiments would never be allowed today. If you work on vertebrates, whether we're talking zebrafish or humans, there's a whole process, in this country at least, that you have to go through to demonstrate that the benefits to society are worth whatever suffering the experimental animals may undergo. It's restricted to vertebrates. You can do anything you want to fruit flies, which is one of the big advantages of fruit flies. Nobody cares what you do to fruit flies. But if it has a vertebrate, whether we're talking frogs, fish, anything with a vertebrate, and with humans, it gets far more complex. There's actually a whole series of forms you have to submit, and it gets evaluated by a committee to determine whether or not the benefits, the potential benefits to society outweigh the harm caused to the experimental animals. And it includes virtually everything, including how the animals at the end are to be euthanized. In the case of humans, Psychologists have to also do this because there's concerns with even pencil and paper uh, test that they could become traumatized. And you have to convince the committee evaluating your proposed research that you are not going to be targeting a subpopulation. That is, you're not going to be focusing on any disadvantaged individuals or anything like that. It's a very complex thing. We've come a long way. As late as the 1950s, 1960s, we were doing experimentation on prisoners on death row and things like that. But today, ethics play a much larger role, as they should, although sometimes it truly is burdensome. First vaccination, the first usage of the term was in 1796. My understanding is that the origins of the word vaccination is related to from bovines, from cows, is where the word came from. There are roughly 200 safe and effective vaccines that have been developed, not just in humans. The number in humans is actually much, much smaller than that, in the orders of tens, not hundreds. We've been unsuccessful with far more attempts at developing a vaccine than we have been successful with. Probably the most famous is attempts to develop a vaccine against HIV. Literally billions of dollars have been spent on attempting to develop a successful vaccine. From my reading of the literature, there are some vaccines that appear to be about 70% effective, but that means 30% of people will contract the HIV and, in the absence of medical treatment, progress to AIDS and death. It illustrates the fact that we don't fully understand the immune response. Common cold is also caused by a coronavirus. We've been trying to develop vaccines for that probably longer than we've been trying to develop them for HIV. And we started trying to develop for HIV in the 1980s. So 40, 50, 60 years, we've been trying to develop an effective vaccine against the common cold with no success. The flu, we don't have a universal vaccine for the flu. Each vaccine is strain specific for the different strains of the flu. And it becomes a guessing game as to which flus are going to arrive on our shores and result in a nationwide infection of people with the flu. The flu is also a coronavirus. And this illustrates the problem with reductionist biology. We know, or we think we know, all of the components that are involved in the immune response, but we know them as individuals. We don't know them as integrated whole, as a complex. And that's what systems biology attempts to do, is to integrate the information. The anti-vaccination movement, if you're not aware of it, started with a physician in Britain at the time who was trying to develop his alternative to common vaccines. And so he published an incredibly small study. I think it's like 4 patients or something like that. It was just terrible science, where he claimed that there was a causative event between vaccination and autism. In particular, he claimed that the MMR, which is vaccination against measles, mumps, and rubella, caused autism. Now, the diagnosis of autism is certainly skyrocketed. It was a term that I don't recall ever hearing when I was growing up. I didn't hear about it until I was probably in graduate school. Autism just really wasn't diagnosed as a separate syndrome back in the 80s, 70s and 80s, 60s, 70s, and 80s. The incidence of diagnosis of autism has gone up, but it's not clear that the incidence of actual autism has gone up. It may be that we're simply diagnosing situations where we largely ignored in the past. It's like, if a state claims that they have busted 5 times as many meth labs as they had in the previous year, does that mean that 5 times as many meth labs were opened, or simply that law enforcement was much more successful in busting these labs? So because you see an increase doesn't actually mean it's a literal increase. It's just like testing for the coronavirus, COVID-19. You're seeing large rates of people that are infected by it. But it has more to do with the fact that we are testing far more people today than the fact that it is also infecting a lot of people. So the numbers are going up for 2 reasons, but a large part of it is because of testing today. This phony, he wasn't a scientist, he was a physician. There really are differences. The study was not reproducible. Nobody could reproduce it. There's been something like 20 or 30 studies that show there is no linkage between vaccination and autism. It's simply a correlation. One of the things you need to be aware of and be alert to is that correlation is not necessarily causation. My favorite correlation that I think disproves the concept that correlation results in something is that all heroin addicts started on milk. It's close to a 100% correlation, except for the poor children that are allergic to milk. 100%. But because you drank milk as a baby does not mean you're going to become a heroin addict. There's lots of other examples, but that's probably my favorite one because I think it's so obvious that it's not causative. It's simply a correlation. And kids are getting vaccinated at a relatively young age, which is also the time that autism can be diagnosed. So there's simply a correlation in time. There's no causative whatsoever. It's been disproved numerous times in numerous studies. Unfortunately, our social media, Facebook, various other things, you have which I'm sure you've heard about, echo chambers, where people insulate themselves in their own silo and they only talk to other people who have the same biases regarding what's going on. And unlike a lot of conspiracy theories, the anti-vaccination group, at least originally, was more liberal thinking. It was people that were anti-pharmaceutical companies. I've talked to some of the people that are that way, and they go on about how, the pharmaceutical company is just trying to get rich by selling us vaccines, blah, 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 blah. They don't understand the economics. You get a vaccine, and many vaccines last a lifetime. So the pharmaceutical company gets one payment for the vaccine. It's not like statins and other things that pharmaceutical companies want you to get on and stay on for the rest of your life. Vaccines are the most economical investment an individual can make. And my wife and I and our son has been vaccinated for lots of things, in large part because we've gone to China and Cuba and other places. Unfortunately, this is a very clear example that at least the United States is no longer an evidence-based society. It's all about fake news, about alternative facts, all these other things. We are no longer an evidence-based society, which is horrible. America became great, not because of golf courses, but because of science and technology, engineering, not alternative facts and things like that. Maybe with the coronavirus, we'll get back to an evidence-based society, which is what we need to be. Systems biology, failure to develop vaccines illustrates the problem with the fact that the reductionist approach is limited. It tells us the components, but it really doesn't tell us how they integrate. Knowing all the parts, as I said, tells us nothing about integration. Systems biology seeks to understand the particular process as a whole. This includes proteins, proteomics, RNAs, transcriptomes, metabolites, other small molecules, hormonal, neural networks. Its attempt to integrate every component of a process. These interactions are very, very complex. I'll show you in a minute one that's relatively simple, but nevertheless very complex. Tools to approach this problem have only recently become available. Large databases, because of research and deposition of data in these large national databases, international as well as national databases, and the ability of computer processing. 20 years ago, we could not really approach systems biology because we didn't have the databases, we didn't have the computing power. Now we can begin to approach this integration of the various different components. First, you need to know all the players, which is very difficult to do because you don't know what you don't know. But you start with various different approaches to try to understand all the different components. Next, you want to know what the interactions are between the different components, what their physical associations are. Do they form a complex? Do they metabolize something? Next, you want to know how do perturbations in one system affect other systems? How can a mutation in a given gene have multiple different effects, pleiotropic effects? What you're seeking in systems biology is emergent properties, properties that arise from the function of a system as a whole, not just the sum of its components, but how they function together in a unified fashion. You're making connections between the various things. And this is a simple illustration. Okay, you need biological data. You need technology, you need computation, and it's a cycle. You're just constantly trying to improve upon hypotheses and understanding of the system. First, scan the literature. It's become so much easier today with computing power and the internet. So you scan the literature, you try to find out all the components that you can find. When I was a graduate student, there was a fairly well-known quote or caution, if you will. Why spend a week in the library looking up things? Instead, you could spend 2 months in the lab repeating something that's already been published. So the first thing is you go to the literature, you find out what's known. Then, after you've acquired all this information, you try to formulate a hypothesis. One, hopefully with a question that you can address experimentally. You integrate the data graphically, mathematically. Graphically is probably for most of us, is the most informative. Then you repeat steps 2 and 3. So scan the literature, look for any new updates, modify your hypothesis based on the results of what you've experimentally demonstrated, and then you go back and you re-examine your hypothesis. You look for new data that may help you to improve the model that you've developed. Ideally, at some point, these models will enable prediction of how the system is going to behave in different circumstances. That is, when you perturb different components of the process. As I said, it requires acquisition of global data sets and integration of different types of data. In yeast, galactose is a very well-studied process. There are 9 genes known to be involved in the biological breakdown and utilization of galactose, all enzymes, as well as the control genes that are involved in regulating it. So it's a relatively straightforward and simple system. We know the different components of what's going on, at least in the metabolism of it. But when you look at how it perturbs other systems when something goes wrong, you can see that perturbing one enzyme in the process results in a tremendous amount of changes. Here you've knocked out one of the controlling genes. The yellow or gold connections indicate protein to DNA connections. The blue ones are protein to protein. And the intensity of the gray indicates the strength of the response. Some very minor, some very strong responses. But you can see in a fairly simple system, about as simple as we can get in a eukaryotic organism, all the ramifications when you try to integrate all the information in systems biology. It's critical. We need to move on to this. It's how we're going to understand how to make effective vaccines in the future. I am personally not optimistic that an effective vaccine is going to be developed anytime in the next several years. As I say, we've been trying to do it for the flu, the common cold, HIV. There's just so many failures because we don't understand the integration of the different components of the immune system. We think we've identified all the components through reductionist biology, but we don't know how they work together to accomplish what they do. There we go. Okay. Perturbations in the cellular network. Prostate cancer is a good example. Roughly 10% of perturbed proteins end up being excreted into the blood. The altered levels of these proteins can reflect the disease state. There is a protein called prostate-specific antigen, PSA, that is often used in the diagnosis of prostate cancer, but it's not very good as a prognostic tool because the results of PSA do not become completely reliable until like stage 3 or 4 of the cancer. By then, it's pretty much too late. There are a lot of different things that can cause an increase of PSA in the bloodstream, many of which have nothing to do with cancer. And so as a diagnostic tool, it's really pretty terrible. There are at least 50 additional proteins whose expression is altered in prostate cancer. And so if we could have an integrated approach to detecting all of these proteins, we might be able to diagnose prostate cancer at earlier stages when it's still much more treatable. Integrated assessment of all the different components would increase the value of these diagnostic tests. PSA, for a while, it was the gold standard and everyone was using it. It was terrible. Physicians knew it was terrible. As I say, it doesn't become reliable until a very advanced stage when PSA levels are actually quite high. Before that, lots of things can cause an increase in PSA in the blood. So it was not terribly reliable. For a while, the medical community said we should no longer use PSA as an indicator because it's pretty much worthless. By the time it's reliable, disease is too far advanced. And now recently, some physicians have gone back to relying upon it, but by and large, it's worthless. But if we could integrate and test these additional 50 proteins, our likelihood of being able to accurately diagnose at an earlier stage would go up. Okay, we have enough time. This is, I believe, one of the best YouTubes I have ever done. It starts out, it's about anti-vaccination, but really most of the lecture is about biases, and biases that many of us are not aware of. And so this one I'm going to play for you guys, hopefully.
I'm Neil deGrasse Tyson, and I want you to know something. If you only watch TV, search internets, and read books that agree with what you think, you have constructed a box. You have boxed yourself. You are in a bubble. The bubble is a sphere, and a box is a cube.
Can you all hear this?
You're in it. If you don't know how other people feel, it'll be very hard to have a conversation with them. You just get into a fight. No, you're right. No, I'm wrong. You're right. If you know how they think, that's a start. It's going to take work. It takes work, especially in these modern times where everybody is so polarized. Take that out and spread the love.
We haven't talked about the vaccine debate here on SciShow because there is no debate to have. Vaccines don't cause autism, and they save millions of lives every year. But there is a debate, whether or not it makes sense. And a lot of people counter this with ridicule, but we at SciShow aren't about judgment, we're about science and using it to better understand the world. We see the anti-vaccination movement as a phenomenon to be understood. So instead of making yet another statement about how yes, vaccines are good and no, they don't cause autism, let's use science to understand why fewer and fewer people are getting their children vaccinated. I'm Hank Green, and this is SciShow. First, let's discuss how we ended up with this imagined link between vaccinations and autism in the first place. Autism diagnoses are definitely on the rise. Now, many scientists believe that this is largely or even completely because of more effective diagnosis and changes in how the diagnosis is reported. So while the diagnoses of autism are increasing, we can't say for sure whether the incidence of autism is also increasing. If it is, it must be because of some environmental factor. Now, when we talk about autism, we're really referring to a range of developmental disorders which can affect a person's ability to communicate or socialize, or cause them to develop patterns of behavior that become pretty specific and inflexible. The condition can manifest itself in a lot of different ways, but you've probably heard of them referred to together as autism spectrum disorder, or ASD. While ASD has been found to have some strong genetic components to it, there also seem to be environmental factors at work as well. And that's really the root of this controversy. We simply don't know precisely what causes autism. And in the absence of explanations, people tried to make make sense of it themselves, and the way our brains do that is almost entirely with cognitive bias. A cognitive bias is really just anything that skews how we process and interpret new information. There are tons of different kinds of bias. Some biases cause us to ignore certain data, others lead us to put too much emphasis on certain data. They can even drive us to focus on facts that are actually irrelevant to what we're observing. But essentially, when we hear a hypothesis and think, yeah, that makes sense, really what we're saying is, yeah, that fits with my cognitive biases. And so people blame all sorts of things for autism: plastics, pesticides, the use of antidepressants during pregnancy, GMOs, sugar, gut bacteria, and vaccines. Basically, you start with whatever makes the most sense to the person doing the hypothesizing. The onset of autism typically happens in one of two ways. Either parents notice a delay in language development, typically around the first birthday, or they notice an apparently sudden loss of existing development, which might happen all the way through the third birthday. Now, humans are pattern recognition machines. We need to be able to figure out what behaviors and strategies lead to positive outcomes. But even more than that, we're on the lookout for things that lead to negative outcomes. This overweighting of negative outcomes is a well-known psychological effect called negativity bias. So imagine you wake up one morning and your car doesn't work. Your brain is gonna want to know what happened. Did you leave your lights on? Did you drive through a huge puddle yesterday and maybe shorted something out? There has to be some reason why it won't start. On the other hand, if you get in a 15-year-old car and it starts up just fine after having had a bad week of barely getting going, you tend not to wonder, what went right? We spend far more cognitive resources attempting to figure out why a bad thing happened than we do trying to determine why something good happened. In psychology, the search for these explanations is called explanatory attribution, and different people have different explanatory styles. Some people are more prone to blame themselves, while others search for an external event to blame. But one thing is clear: we are very bad at not blaming anything. It's not surprising that parents of children with autism, especially parents who notice a sudden loss of previous development, will search for a possible cause. And when the most significant recent event in the health of a child was a vaccination, as can be said for many moments in the life of a young American, we might identify that as a potential cause and deem that link worthy of further examination. Now, this is completely logical. The problem is that over a dozen peer-reviewed papers have found no correlation between autism and the MMR vaccine, or any other vaccine for that matter. And yet, when you Google vaccines and autism, a fair number of the results claim that there is a link between the two, and that link is being covered up either by the government or by big corporations. A parent already experiencing frustration with the medical community's inability to tell them why this thing has happened to their child will, on the internet, find a vibrant community of similarly frustrated people who share their values and experiences. These communities are full of anecdotes that draw connections between vaccines and autism, and so unsurprisingly, some people become convinced that they have found the reason for their child's disability. Once their mind has been made up, confirmation bias sets in. Confirmation bias is simply our tendency to more readily and with less scrutiny accept information, anecdotes, and worldviews that confirm our existing beliefs. And again, it is a completely normal thing that every person does. Indeed, trying to convince someone that a previously held belief is incorrect has been proven to actually increase their affinity for that idea. And so a community is born, and the safety of vaccines is called into question. And once the procedure for getting a vaccine goes from the doctor telling you that it is now time for a vaccine, and 99% of parents agreeing because that person went through medical school, to it being a question to ponder, vaccination rates will go down. A 2011 study showed that parents who think about vaccines before their child is born are 8 times less likely to vaccinate their children. Basically, when given an opportunity to do research on their own, what they find is confusing. And when confused, the default choice is to simply take no action. And this is an example of yet another bias called omission bias. In effect, we judge harmful actions as less moral than harmful inactions or omissions. In fact, a frequently cited study found that when the choice to vaccinate is framed as an action, the average parent will only vaccinate their child if not vaccinating is at least 2 times more dangerous than vaccinating. This has to do with our perception of future regret. Parents report that they'll feel worse if they take an action and it harms their child than if they don't act and the child is harmed by a failure to act. This perception of potential regret can be so strong that even bringing up the choice of acting versus not acting seems to be counterproductive. A 2013 study found that attempts to convince parents to vaccinate their children actually decreased the percentage who went on to choose vaccination. If vaccination is presented as a personal choice instead of a necessity for good public health, then potentially harmful inaction can seem more moral than potentially harmful action, and vaccination rates go down. Parents are choosing to let nature take its course. And as you might expect, this effect is much stronger in people with a measurable naturalness bias. This is just a tendency to perceive things that come from nature as being inherently less threatening than things that we invent ourselves. themselves. One way psychologists measure this bias is by asking a subject if they'd prefer a substance extracted from an herb or one synthesized in a lab, even if they're chemically identical. And of course, others have biases against big government or big corporations, and these ideas about vaccinations fit well with those worldviews. Confirmation bias at work again. But even people who don't hold these biases end up being more likely not to vaccinate if they start doing research before their baby is born. This is because of another failure of the human brain. We are terrible at what psychologists call risk perception. Given the merest sliver of a possibility that vaccines will cause developmental disorders, parents are now weighing a disease they have seen, autism, against diseases they have never seen. Since the 1970s, measles has been pretty much unheard of. The measles doesn't scare people my age for the same reason a giant man-eating squirrel doesn't scare us. We've never seen it. Risk perception is basically a science all on its own, and we've found that vague future hazards, like the future probability of an illness, are far less frightening than immediate specific hazards, like the sudden onset of autism. So amazingly, the success of vaccines is one of the reasons that people are less likely to vaccinate their children. So yes, it turns out humans are complicated, and this is a complicated problem. Humans are inherently bad at understanding the effects of self-selecting samples like online anti-vaccine forums, and often completely unable to accept that a negative outcome could really be the result of something that's beyond their control, and still not very well understood. This is not an anti-vaxxer problem. It's a human problem. Those of us who trust science or have built an understanding of statistics and bias simply have had different lives than people who more heavily weight anecdotes or the opinions of their friends or strangers they meet online who feel the same way. So next time you find yourself frustrated about the decline in vaccinations in America, remember that it's only because of the dramatic success of vaccines that we could even think of having this debate. And those anti-vaccine activists are being driven by the exact same logic traps and cognitive biases that every one of us suffers from. Only by understanding and accepting these psychological pitfalls that we're all so susceptible to will we be able to solve this problem, and that is what science is all about. Thanks for watching this episode of SciShow, where we really do try to be objective, and we objectively believe that the universe is amazing and fantastic, and if you want to join us in understanding it and all of the stuff in it, including our brains, you can go to youtube.com/SciShow and subscribe.
Okay, I hope you all found that informative. I think it's exceptional. I would encourage you to share it with friends, and bear it in mind when you're talking with people whose views may differ from yours. I want to leave you with one sort of personal story, but it's meant to illustrate that you should never be afraid to ask. My wife was a computer programmer. She went from being a receptionist in the computing center at UVA to supervising computer programmers by the time we left there. Her degree is actually in Parks and Recreation from the Davis College at WVU. We both thought we wanted to be park rangers. That didn't work out for either of us. In desperation, she took a job as a receptionist. She talked people into giving her small projects and allowed her to take courses in computer science. And eventually she became a supervisor, and she was the assistant director of information technology here. So she took advantage of opportunities. And many of you are going to be facing graduation, if not this year, then next year. And it's going to be a stressful time. It is for everybody. But don't be afraid to take opportunities. It doesn't have to be related to what your degree is in. She, as I said, was a computer programmer. She was heavily involved in when the university implemented a software data analysis system known as Oracle. It keeps track of tuition and, how many females we have, how many, just everything you can think about. It tracks it for the whole university. It was a very demanding task to get this whole thing implemented. But Oracle, which is an extremely rich company, and if you're a fan of Iron Man movies, I think the second Iron Man movie, Oracle has a big sign up right in front of one of Iron Man's suits. Oracle also has sponsored the America's Cup for the last decade or so. It's a very wealthy company. Each year they have an annual meeting that occurs in San Francisco. Most San Franciscans hate it because anywhere from a quarter to a third of the city gets shut down by so many people showing up. It's an amazing conference. I've got to go with her a couple of times and managed to get passes to be part of it. They literally, they have, like most conferences, they show products. Salespeople come up and they show products and this, that, and the other. And a lot of them, they give a lot of different things away. Well, one year, everyone who went to the conference actually got a laptop or a tablet, got a free tablet. Another time they got leather backpacks. They would literally be auctioning off BMW motorcycles. It's just the most expensive, elaborate, complex conference I have ever been to. I go to a fly conference, I get a ticket that enables me to get a beer or a Coke. That's it. There, amazing food, just an amazing organization. But we went there one time, and I'm not a huge fan of San Francisco, actually. I'm not sure why so many people are enamored with it. The culture is amazing, the food is amazing, but otherwise, scenery and everything, I don't think is that great. But I went there one time and I thought, well, I've never heard so much about Alcatraz. I've never been there. And so, well, this didn't work out the way it was supposed to. And so I decided to go. And this little box is actually supposed to be covering him. But anyway, this couple was talking to a park ranger. And it looked to me like they were initiating a tour. And so I thought, well, what the hell? So I walked up and I asked, I said, looks like this is a tour that's starting. Could I join the tour? And the park ranger was delighted to have me join the tour. Now, this is before I could see this guy's jacket. So the three of us and the park ranger, we started on a tour of Alcatraz. It turns out this guy, who's a Hell's Angel, is a huge fan of Al Capone. Somewhere on his body, he has Al Capone's face tattooed on him. I don't know where. I don't want to know where. But the park ranger was delighted to have me along. He was obviously nervous. The normal tour of Alcatraz, if you haven't been there, lasts about 20 minutes. The tour I was on lasted about an hour and a half. We went into places where we had to wear hard hats because of the concerns about falling debris and whatnot. We went to every place that had a connection to Al Capone. The San Francisco police, or the California State Police, I'm not sure which, I can't remember now, asked Alcatraz to give this guy a personalized tour. I've got to think he was some kind of snitch or something, I don't know. But we went everywhere. All because, hey, I asked, can I join you? And we did. We went everywhere. And the park warden, he showed us how the gates shut, and both the male and the female visibly shuddered. They obviously had heard that sound before, and they said, please don't do that again. But it was interesting. So I got a heck of a nice tour because I asked. So don't be afraid to ask. Don't be afraid to follow paths that may not relate to whatever your degree is. Just take opportunities as they come to you. Anyway, good luck. I will be posting this on YouTube. The way to search for it is to look for Bio324, all as one word. You can do WView, Bio324. There are now, currently, there are 7 postings. Some of them are short, some of them are long. Some of them have a long dead time at the beginning because I logged in too early to Zoom, and I apologize for that. So some of them have like an 8 or 10 minute dead time at the beginning. But then it goes through the whole thing. So I haven't been dropped this time, so this is going to be one of the longer ones. I will have it uploaded so that you can view this if you so wish. I will be sending you emails, updates. The exam will be, the third exam will be April 30th. Currently, the final exam, for those of you electing to take it, will be the following Monday. I am going to work very hard to get at least the exams graded for people that are on the border between different grades, and get it back to you in time. Please, as I indicated in my email, do not turn it into a PDF or anything else. Just leave it in Word. It's so much easier for me to grade it if it stays in Word and write comments. And also, as I found out, if you could add your last name, and there's only 2 people in the whole class whose last name I know. Add it onto the document, the name of the document. It helps me to organize things much more efficiently. So next time we meet, coming Thursday, or is today Thursday? God, I've lost track of the days. It's terrible. Next time we meet, we're going to talk about metamorphosis. It's going to be a very abbreviated presentation. It's what I think is one of the more fascinating things about biology. And we're going to focus primarily on amphibian metamorphosis and why it's such a fascinating event. I will send you the key to the practice exam. If you have any questions, feel free to send me an email and I will respond. Stay healthy, stay safe, and see you next class period. Bye.